back and we're going to finish up this chapter by looking at polysaccharides. So we started with monosaccharides, disaccharides, and now we're going to finish with polysaccharides. So these are many sugars that are linked together. So you will have, in this case, similar types of naming that we saw with disaccharides. So again, you have those glycosidic linkages, whether they're alpha or beta, and what carbons they're connected to. You just repeat that sequence over and over and over again. And there are different types of polysaccharides. The two main types that we will see in this chapter are what are called homopolysaccharides. That means it's the same sugar and the same glycosidic linkage, just repeated over and over and over again. We also have heteropolysaccharides. That's different. So we have different sugars that are just repeated, right? So we have, instead of just one sugar only, you might have repeats of two sugars over and over and over again. A galactose and a glucose, a galactose and a glucose, a galactose and a glucose. But they have the same glycosidic linkage throughout. So maybe you've heard of things like people taking glucosamine and chondroitin for their joints. Chondroitin is a type of heteropolysaccharides. Heteropolysaccharides are really good with cushioning material and cartilage and things like this. Whereas homopolysaccharides we use for energy storage or for structural capabilities, different things like this. Again, different types of sugars serve different types of purposes. So let's take a look at these polysaccharides a little bit. So if you take a look, starches, okay, so there's two types of polysaccharides. Again, many different sugars together, depends on the types of sugars and the types of linkages. Now, if you're looking for linkages, alpha linkages are weaker, which means they're easier to break, which means we use these for energy. These are energy polysaccharides. These are things like glycogen, okay? So alpha linkages are common in things like glycogen or what we call amylose, or you may have heard of amylopectin, or if you are into looking at jellies and jams and things like this, you might have heard of pectin before. But if you notice, if you make jellies and jams, it's not solid, but it's not liquid. It's kind of that, in between, and you can get more movement or less movement, and it depends on that type of, how much energy is in those linkages to make them more structural or less structural, which we'll talk about in just a minute. So amylose, okay, which is what we're going to start with first. Remember, ose, we can look at sugar. Amylose is a homopolysaccharide. So again, that means the same sugar over and over and over again, just repeated over and over and over again, more than two, many, okay? It's a homopolysaccharide of glucose, alpha, 1,4 linkages. So you have a glucose, alpha, meaning opposite, carbon 1 bound to carbon 4, and you repeat that over and over and over and over again. Now, I would not have you draw 16 different glucoses all connected. But instead, you should be able to determine what it would look like and pick that out. So as you can see here in the slides from your book, here we have a glucose, and a glucose. If you just look, you have alpha because the OH is going down, opposite of the OH, carbon number 1 to carbon number 4, carbon number 1 to carbon number 4. So you just repeat that same thing over and over again. That's it. So that's what's kind of neat. So if you've ever noticed, maybe you've heard of this before, there's an enzyme, which we'll talk about enzymes in chapter 16, called salivary amylase. Okay? You notice when we say ose for sugars, ACE is for enzymes. So salivary amylase is an enzyme that breaks down amylose. So this is if you've ever tried the saltine cracker challenge, I guess. If you take a bit of that saltine and put it on your tongue and leave it there, it'll turn from salty to sweet. And that's because the salivary amylase starts breaking down the starch, the very long polysaccharide, okay, breaks it down into the single or disaccharides of sugars, which are sweet tasting to us. So you can try that if you would like to, okay? So you can try that saltine experiment if you would like. So that's amylose. It's just glucose 1,4 over and over and over again. However, if you have glycogen or amylopectin, Glycogen and amylopectin have what we call branches. So you have, I'm not going to draw 15 different sugars over and over again, but let's just pretend this straight line here is glucose alpha 1, 4, right? So you have your carbon 4 here attached to your carbon 1. But now we have carbon 6. You might have other branches of alpha 1, 4, but there's an attachment here between carbon 1 and carbon 6, as you can see from the better looking structures in your book. So if you look here, here's your alpha 1, 4 all the way through. But now here's carbon number 6. This OH group now is attached alpha, because again, this OH group up, this OH group down. This is an alpha 1, 6 glycosidic linkage. So you have one homopolysaccharide, just alpha 1, 4, but then you have branches that go off of carbon 6, but they continue as alpha 1, 4, alpha 1, 4, alpha 1, 4. Now, when we store glycogen, glycogen has a lot of these, so amylose has no branches, has no 1, 6 linkages. Glycogen has lots of these branches. It prevents it from packing together. It also gives it more ends for our enzymes to access, so that when you all of a sudden start exercising or start moving, when you don't have enough food and glucose in you, you can use glycogen, but you have to trim from the ends. Well, if you only have amylose, you literally have 2 ends, that's it. But if you have glycogen and you have all these branches, now you have all of these different ends that you can grab from. So the more branching that you have, the more ends that you have, that means you can get quicker energy to do what you need to do. So that's what we're going to talk about for starches. So starches are alpha linkages. They are what we use for energy. When you have beta linkages look at more what we call structural polysaccharides. A beta linkage is a stronger linkage. Okay? These are more what we call structural polysaccharides. So a great example is cellulose, just like we talked about in trees. If you think about trees, tree barks, trees are very strong. Beta linkages are stronger linkages. If you're interested in knowing more, please let me know. I can explain it. But cellulose and amylose are the same chemical constitution. The only difference is the OH group going up or is the OH group going down? That's the only difference. Okay? But one of them, trees, one, energy for us in our muscles. Only difference, alpha, opposite, or beta, same. That's the only thing that's different between the two. Okay? So amylose and cellulose are the same exact thing. It's just whether is the OH group up or down, same or different. Chitin is another example. You may have heard of chitin before. It's what we see in exoskeletons of different bugs and things like this. It's very hard outer shell, again, structural polysaccharides. But in this case, it's something called an N-acetylglucosamine. So glucose is in there, but you also have that amine group with that nitrogen that's on there with an N-acetyl group. And again, humans, we cannot break down, we don't have the ability anymore to break down these beta linkages, which is why some organisms, right, do have the ability to break these down. A lot of, vegetarian species that feed on tree bark or things like this, they might have the ability to break down those beta linkages. We, as humans, do not have that ability anymore. So if we take a look, right, so now that we know about polysaccharides, that's important because we can link these polysaccharides or oligosaccharides on the outside of cells. Now, we don't just want to have a single sugar usually, because that doesn't give us variability. But if we have, 2 glucoses and a galactose, and then we can switch out this galactose for another glucose or a xylose or a erythrose or whatever it may be, you can give a bunch of different stereochemistry to make your sugars very diverse. And that's, again, goes back to that whole communication between cells, viral entry. If you just had one receptor and one sugar and that was it, everything could get in and everything could get out. You wouldn't be able to tailor the needs of the cell to things coming in and out. Okay? But again, we can also put that on the outside of the cell. That's a good way for our red blood cells, for example, to recognize us based on our blood types. So again, glucose, galactose, very different receptors, and different combinations of those give us the huge variety to recognize a whole bunch of different sugars. So here, this just shows you from your book, here's like a membrane. Here is your sugar sticking on the outside of the cell to receive a signal to transduce that inside the cell to say, hey, you should do more of this, or you should make more of this, or you should get more of this, or whatever that may be. So cell markers are a really great place to do that. So if you take a look, this is the final thing about how important carbohydrates are. So the difference between A blood group, B blood group, and O blood group is literally the difference in sugars. So this just shows you an example that if you have an O blood group, you have each of these little hexagons here represents a sugar. So everybody, if you notice O, you may have heard of O being the universal donor. So each of these blood groups, right, whether it's A, B, or O, if you notice, they all have the same 1, 2, 3, 4, 5 sugars. So O blood group doesn't, the only difference is A and B have a 6th sugar. Remember, these on the outside of your red blood cells. So if you take a look, there's no additional sugar on the O blood group. I know I say no sugar here, but I'm talking about that additional sugar on that O blood group. If you're an A blood group, you have an extra sugar at the end, and it's called an N-acetyl-galactosamine. So the parent sugar there is a galactose with an additional nitrogen and 2 other carbons. If you have B blood group, it's just a galactose, right, which is a diastereomer of glucose. So again, if you give somebody the wrong blood type, right, if you are O and you receive B blood, your body will be like, this isn't us, and it's just because of that one sugar. And again, so hopefully from this chapter, I've convinced you that knowing about sugar biochemistry and understanding those seems like very small differences, glucose versus galactose, enantiomer, diastereomer, or alpha versus beta, they might seem like small differences, but those small differences make a very big difference in us.